is pangenomic. Basically, we will discuss about plant pangenomic. Here are the content of this presentation, and next chart is the introduction. Pangenomic is a genomes of multiple individual cultivar population. Thus, a pangenome represents the entire set of genes within a species. The field of study of pangenome is called pangenomics. Meaning everything, and genome meaning complete genetic material. It is defined by Tillian in 2005. It comprises of two portions: core genome and dispensable genome. Core genome consists of genomic regions or sequences that are conserved throughout the individuals or group of that species. In short, it contains genes present in all strains. The remaining portion of pangenome, which is not conserved and show lot of variation, are composed of genes absent from one or more strains and that are unique to each strain, is termed as dispensable genome. Here is the picture which shows core genome, unique genome, and a dispensable genome of three different cultivars. Here is the types of pangenome. Number one is closed genome, and second is the open genome. Closed genome is those genomes which contain large core genome and small accessory genome, while open genome contain small core genome and large accessory genomes. Objective of this is the comparison of traditional genomics and pangenomic approaches. Discuss about the methodologies of plant pangenome, growth of the application of plant pangenomes. Here is the comparison between traditional genomes and pangenomics. Traditional genome typically focus on sequencing, assembly, and annotation of a single reference genome for a species, while pangenome explains beyond the scope of single reference genome to encompass the genetic diversity present across multiple individual or varieties within a species. Next is the methodology. Methodology of traditional genomics relies on sequencing technologies and bioinformatics tools to generate and analyze the genome sequence of single individual, while in pangenome it employs similar sequencing and bioinformatic methods but extend them to analyze multiple genome within a species. Objective of traditional genomics aims to characterize the genetic makeup of species by identifying genes, regulatory element, and functional element in a reference genome, while in pangenomics it seeks to go beyond the reference genome to explore the genetic diversity and evolutionary dynamics within a species. Application of traditional genome has in gene diversity, gene discovery, comparative genomics, and understanding the genetic basis of trait, while pangenomics has application in crop improvement, biodiversity conservation, and evolutionary studies. Moving forward is the methodologies of plant pangenomics. Number one point is the genome sequencing, whole genome sequencing of multiple individual or variation varieties within a species from the foundation of plant pangenomics. Next generation sequencing technologies such as Luminase sequencing, PAC biosequencing, and Oxford nanopore sequencing are commonly employed for this purpose. Next step is the assembly and alignment. After sequencing, the raw data are processed to assemble individual genome. This involves aligning short sequence to a reference genome or de novo assembly to construct whole genome sequences. Next step is the pangenome construction. Once individual genomes are assembled, the pangenome is constructed by integrating and comparing the genomic sequences. Next step is the annotation and functional analysis. Genomic sequences are annotated to identify genes, regulatory elements, and other functional elements. Next step is the structure variation analysis. Plant pangenomic involves the identification and characterization of structural variations such as insertion, deletion, duplication, inversion, and translocation across the genomes of different individuals. Next step is population genetics and phylogenomics. Population genetics analysis are conducted to study the genetic diversity, population structure, and evolutionary relationship among different individuals or populations within a species. Second last step is the association studies. Pangenomics data are utilized for genome-wide association studies to identify genetic variants associated with specific traits such as disease, resistance, yield, and quality traits. Last step is the pangenome visualization. Visualization tools and software are employed to visualize and interpret pangenomic data effectively. Genome browsers, comparative genomics viewers, and interactive visualization platforms help researchers explore and analyze large-scale genomic database. Overall, plant pangenomics integrates various genome, computational, and statistical methodologies to comprehensively analyze the genetic diversity, evolutionary dynamics, and functional implications within a plants. Here is a picture which shows how is plant pangenome generated. And next are the applications of plant pangenomics. Number one is the crop improvement and breeding. Plant pangenomics provide valuable insights into the genetic diversity present within a crop species. Next is the understanding genetic variation and adaptation. By analyzing the plant pangenome of plant species, researchers can cover the full spectrum of genetic variation, including core genes shared across all individuals and accessory genes specific to certain populations or ecotypes. Next is the functional genomics and gene discovery. Plant pangenome facilitate the identification and characterization of novel gene regulatory element and functional variant that contribute to important agronomic traits. Next is the disease resistant and pathogen interaction. Plant pangenome analysis to plant pathogen interaction enable the identification of genetic factors associated with disease resistance and susceptibility. Next is the phylogenomics and evolutionary studies. Plant pangenomics provide insight into the evolutionary history, speciation event, and divergence pattern within the plant species and genera. Next is conservation and biodiversity assessment. Plant pangenomic studies contribute to the conservation and management of plant genetic resources by identifying genomic variation within wild and cultivated population. Last is the crop wild relative and integration analysis of plant pangenome data from crop wild relative help identify valuable genetic trait for crop improvement throughout integration breeding. Overall, plant pangenome offers a powerful framework for understanding the genetic basis of plant diversity, adaptation, and trait evolution with broad application in crop improvement, biodiversity conservation, and sustainable agriculture. Last is the conclusion. Plant pangenome provide a holistic understanding of genetic diversity within species. It reveals core genomic elements shared among individuals as well as accessory components specific to certain population, informing breeding and conservation strategies. Plant pangenomic analysis facilitate the identification of novel gene, adaptive trait, and genetic variants crucial for crop improvement and ecosystem resilience. By integrating plant pangenome insights, researchers can develop sustainable agriculture practices, conserve biodiversity hotspots, and harness plant genetic resources for global challenges. Embracing plant pangenome fosters collaboration among scientists, breeders, polymarkers to drive interdisciplinary solutions for food security, ecosystem resilience, and human well-being. Thank you.